A very good afternoon to one and all, dear judges and the respective teachers. So as we know, nutrition deficiency is one of the most major crises in this current generation or in this current world. So to overcome or to check this crisis, here I have a mere solution or a prototype and a good initiative to solve nutrition deficiency in human beings and other organisms. So especially in this model, I have used the concept which has the biological background, which is called BIOFROTIFIER. Bios means life and fortification means strengthening it. So BIOFROTIFIER, what do we mean by BIOFROTIFIER? It is a biological process to increase the nutrient level in the plant's byproduct or the human essential nutrients in the plant's byproduct by transgenic mutation, conventional breeding or selective breeding, and the traditional method and agronomic practices. So iron, zinc, beta carotene or provitamin A and the amino acids and the proteins has the scarcity in the staple crops such as rice and wheat and beans and sweet potatoes. So we can use this initiative in that crops. In India, most of the families are consuming rice and wheat as the staple food. So the iron, here I am conducting the following biofortification, or the specificity of the biofortification is listed down. Iron biofortification, zinc biofortification, beta carotene or provitamin A biofortification, and other essential amino acids and protein biofortification. So friends and judges, I am starting to explain or elaborating the project or the model. So here there are different units, as we know. There are numerous units which contribute for the entire process. The entire thing starts from here. So please concentrate here. Here the residue or the plant required product is collected manually. Then this is transferred to this unit. And here I have a motor with the propeller which crushes the content, which is very much required. And the entire project is a biotechnological based project. So this content is contained in these units, and here certain biotechnological enzymes are used, such as cellulase. Here there is a specificity of plant cell, and as we know, plant cell has an additional layer of cell wall, which is mostly made up of cellulose, which is a polysaccharide or a complex sugar. So to digest or to destroy or to break the cell wall, we have to use a specific enzyme called cellulase. For cellulose, we have to use cellulase to break that cell wall. Okay. Then after breaking the cell wall, we have to use restriction endonuclease to break the DNA or the germ plasm. For your kind information, germ plasm is the genetic content of the cell. So to break the DNA, here we want the DNA and we have to isolate it further. So to break the DNA, we have to use restriction endonuclease, which is an enzyme mostly used in biotechnological labs, and it is the most important criteria of a project. And when the DNA is break, then we have to inject it manually or by retroviruses or by bacteria in the host for the multiplication. Okay, then when we injected it manually or by retroviruses or bacteria, the cell will start to replicate in that plant. So we have to screen. We have to see the results of the test. So we can use the blue-white screening especially to check the mutation, and PCR, where the DNA recombination is used. So DNA polymerase is also used in the screening basically. So that is what the thing is. So I am saying the process how this stuff works. So the content is crushed here, then they are collected in these units. Here in this compartments I have enzymes, cellulase, restriction endonuclease, and other enzymes for the functional part of the project. Then this content travels, or this things travel to the DNA recombinator. So this is how this thing works. So I'm stopping it. And as we know, under critical condition, microorganisms mutate themselves. And here, the genomic process or the genomic approach or the mutagenesis approach toward the biofructification is used. And my project name is, for this reason, my project name is the modern biofructifier. Basically, it is a fructifier. So under UV light, it comes below it comes below the visible spectrum. It is highly injurious to us. And under critical condition, the bacteria or the organism mutate themselves, the plant cell especially. So this is the UV rays. This is one of the most essential thing in my project. So the most important or the essential parts are completed. Then these things are stored here. These things are stored here for the screening and other things. Here the things are injected or transferred by modern methods, by retroviruses and bacteria. So this is how the entire thing works. And there is some specification and features for a specific kind of nutrient for iron biofructification. And starting iron biofructification. As we know, reduction in iron causes anemia, and there will be a scarcity of hemoglobin. As we know, hemoglobin is a quaternary structured protein which has two alpha rings and two beta rings, where 6 molecules of iron are contained. So the scarcity of iron will cause scarcity of hemoglobin, and the person will get anemic, and there will be a reduction in the production of the RBC or erythrocytes, red blood cells, and there will be respiration problems also. So in soil, the most abundant or the nutrient which is present mostly is iron, but a specific less amount of iron can be consumed by the plant in specific compounds. So iron or certain specific compounds of iron are biofructified in the soil. This will check the iron deficiency in the plant's byproduct. The zinc and the protein or provitamin A deficiency is also done in this method. There are certain compounds which can be consumed by the plant easily. So this is what the specification and the features, and this can be a good initiative for the government.